Okay, welcome for the last session of today, everybody. And it's a pleasure to introduce Professor K. VijayRaghavan, who doesn't need introduction, but I'll do it briefly. So he's one of the foremost Drosophila biologists working on neurogenesis and myogenesis, which is, I think, what he'll be talking about today. Former director in CPS, former DPT secretary, former principal scientific advisor to the government of India, Padma Shri, Infosys Prize winner, Royal Society fellow, and so on and so on. I met him in, I think, 2004 when I was 31 and unknown to the biology community in India. I had done my PhD in physics, and he met me at some meeting in Mahabaleshwar and immediately reached out for a possible collaboration, which happened. And that's the kind of person he is. In 2012, when we were talking about setting up a computational biology group at IMSC, he was extremely supportive and helpful in many ways. And as DPT secretary and as PSA, he was engaged with the public via social media as well as via private channels. And though not all inputs could be acted on, I think all were read and engaged with. And he was in the hot seat during COVID, and despite the difficulty of herding cats and so on, he made a lot of important contributions, like ensuring some level of open data sharing and so on, and ensuring some scientific input into what drugs were prescribed, things like that. So anyway, without further ado, here he is.
Yeah. Thank you very much, Rahul. It's a great pleasure to be here. There's only one thing worse than being an after-dinner speaker, and that's being a before-dinner speaker. Before dinner, all of you are awake and can throw metaphorical tomatoes and eggs at me. After dinner, that wouldn't be a problem. So congratulations on your 60 years. It's really wonderful to be here. I enjoyed the talks all of today. And I must say, my interaction with mathematicians and theoretical physicists has a history which goes back to my time as a student and faculty member at TIFR. It was absolutely wonderful. The physicists could see through biologists, so they didn't think too much of them. I remember B.V. Srikantan telling Pabitra Maitra when Maitra accosted Srikantan about some serious problem, that Pabitra, you will not understand, you're a biologist. But the mathematicians, because they knew less of biology, had no prejudices about biologists, and so we became good friends with the mathematicians. Not that we were not friends with the physicists. And I remember being very impressed by seeing M.S. Narasimhan writing in the air, and even more impressed by C.S. Sheshadri writing in the air and then erasing it. So these were very impressive people. Balu talked to us this morning about Ramachandra. I was in the hostel at the same time as Balu, and sometimes he would be at the next stall in the shower, and Ramachandra would be outside the door, and they would be conversing. And this was not a irregular feature. It's quite common because it happened many times. So the mathematicians were very interesting, and it's really wonderful to see how math science has grown and the area of science in Chennai in general has grown, and we'll learn more about that. So on to my talk. I'll be talking about cells, circuits, and the developing brain. And really the principal issue which I'll try to address is how ours function they do. But if you look at, can you dim some of these lights here, if possible, but otherwise don't worry. Because some of the other figures may be a little better with lower light in the front. So, if you look at a tiny bacterial cell, such as that of a well-studied bacterium, the Escherichia coli shown here, it's about 1 cubic micron. And the volume of the cell body of a typical neuron, a nerve cell, is about 12,000 times more than that. And in a motor nerve, such as what we have, or elephants, or dogs, or cats have to control our muscles, the axon itself can stretch up to a meter or so without any problem. So the issue is that this nerve cell is extraordinarily complex, much larger than a bacterial cell, and we know rather little about bacterial cells in many ways. They sense the environment just like a nerve cell does and responds to it, but our ability to predict what the bacterial cells can do is not high, despite a lot of research. I mean, we can clamp all inputs and vary only one and see what happens, but if you start varying multiple inputs, our predictability goes down substantially. It's not that, it's only a problem that nerve cells, for example, have interactions with other cells. Our cells and other cells in our body interact with multiple cells. Bacterial cells also do that, and that's been studied very nicely. Groups of cells will communicate with each other. A small number of cells will tell more bacterial cells what to do as a community and what not to do. So cell-cell communication on scale is not just unique to organisms such as us, it's there in multiple other, even in single cellular organisms. So despite this, as I said, studies of over 100 years have, and how cells respond to all kinds of environments, different kinds of cells, have dramatically increased our understanding of the processes involved. So at least at a descriptive level, we know a lot. And at a mechanistic level, we know a lot about the components. Yet, it would be fair to say we are not fully able to move from understanding components of the complexity to be able to predict responses to multimodal inputs. That is when many changing parameters are simultaneously sensed by a cell. So, these kinds of situations, if they're so difficult, the question then arises, how then can we understand a structure such as the human brain? And the human brain, of course, and all the wonderful things that humans seem to be able to do, but also how, for example, other questions such as how the mind relates to the brain. To answer this, and not just leave it as a rhetorical question, we must appreciate the complexity of the human brain and then see how it can be teased apart into components which we can address. Our brains have about 85 billion nerve cells, and they make about 1 trillion connections. While different parts of the brain have specialized function for vision, smell, motor action, and so on and so forth, the parts also communicate with each other, and they do it at multiple layers. So, there's an architecture issue and recursive interaction issue and so on. So there's an enormous amount of input information being analyzed and reanalyzed and fed back at multiple layers. So it's a very complex computational problem as also an anatomical and a physiological problem. So how will our understanding of the brain improve? And Sidney Brenner, one of the most brilliant biologists of our recent times, said that biologists, no matter what the problem is, they ask only 3 questions. That's okay, that's fine. It's just a blank slide. Yeah. So I was told about how to give talks that it's good to have a blank slide so that you can concentrate on what I'm saying and not on what's there. That it always then results in the IT people running up to fix the problem. So it's not good advice. So Sidney Brenner said that, there are typically biologists ask only 3 questions. Where did it come from? That's evolution. How is it made? That's developmental biology. And how does it work? That's physiology, biochemistry, and so on. Of course, if you're a medical doctor, you ask, how do you fix it when it's broke? So our ability to do these 3 things has changed dramatically, particularly in the last decade or so, but over the last 2 or 3 decades, enormously. So our ability to collect data at every level, what the genes are, where they're expressed, how the genes vary, how different components are made by genes, how gene regulation takes place, metabolites in every cell at every time, how connections are made, and so on and so forth. There's an enormous amount of data which is coming in, and that kind of data far, far exceeds what we're collecting from, for example, astronomy and high energy physics and so on. So this is a very big challenge where we're flooded with data and we're short of theory and we're short of concepts. So that's an important thing going forward. So let's go through the first part. Where are we from? Where is the brain from? Where is it from? Now the Earth was born a little over 4.5 billion years ago, and the Sun and the solar system was born a little earlier, only a little earlier, amazingly. And amazingly, life on Earth originated not too long after the Earth was born. We don't know yet how life on Earth originated, and that's a very important problem. There are very interesting theories and experiments going on. But we do know that about 3.5 to 3.8 billion years ago, simple cells, what are called prokaryotes, came to being. And about 3 billion years ago, there was photosynthesis, the ability to use light to generate energy, the sunlight. 2 billion years ago, complex cells, more complex cells resulted, what we call eukaryotes. And about 1 billion years ago, and maybe the dates are being pushed back now, multicellular organisms consisting of more than one cell, multiple cells originated. So it's, from 3.5 billion years to 1 billion years, amazing changes took place. Going further, 600 million years ago, we had simple animals. Then you had more complex animals, about 570 arthropods. There's a huge explosion of complexity. And we don't know how from these simple multicellular organisms to complex organisms, mechanistically what happened which allowed this dramatic explosion. There are lots of theories. And then you had fish and amphibians and so on and so forth. And about 400 million years ago, you had insects and plants. And about 360 million years ago, amphibians. About 130 million years ago, flowering plants. So the world was changing dramatically. Then 65 million years ago, something very interesting happened. We think an asteroid crashed or a meteorite crashed on Earth near Mexico. And that really resulted in a dramatic change in the landscape of life on this planet, eliminating dinosaurs and many large animals, and leading to the beginning of this dominance of mammals on this planet. So 65 million years, non-avian dinosaurs died out. 2.5 million years ago was the appearance of Homo, and about 200,000 years it's since the appearance of modern human, and only 25,000 years ago Neanderthals died out. Now, as a consequence of our evolution, and then as a consequence beginning sometime maybe about 10,000, 50,000, 100,000 years ago, in that range, many things happened. Early on in our evolution, we got the ability to oppose thumb and forefinger, make tools. And then as our brains grew disproportionately to our body size, or at least were over-designed compared to what our bodies immediately needed, we evolved the ability to speak and link speech and language to cultural activities and functions. So sociological and cultural evolution rapidly overtook biological evolution. So today, we are in a situation, not only do humans understand nature's engineering substantially and keep doing that, but we have become the engineers of nature. And we today, as philosophers have said, wield the paintbrush on the planet of the Earth, on this planet, and hold the planet's future in our hands, not just our future. And therefore, we have become, we do things which it's impossible for other animals to do, such as go to outer space and so on and so forth. So the software and hardware of our brains, of course, is impressive, and our cultural evolution is extraordinarily impressive. So when we ask where all this came from, that's evolution. But we can also ask, how did this happen? And here, the common origin of all life on Earth, as far as we know, all life on Earth has a common origin. Chemistry, and it means that shared, and we know now that shared chemistry is tied together by the string of DNA. And as you heard these talks in biology just now, you can study any organism and have lessons for any other organisms because of this shared thread of chemistry and the way DNA ties it together. So how is it made if all this is tied together by the shared chemistry? There, it's actually very interesting. Because of the shared chemistry, there's also a shared toolkit. And rather than study to functions, different tissues and humans are made, biologists typically study simpler organisms and study brains in much simpler organisms. Now, that has resulted in getting enormous information which can be applied to other organisms. So here's a simpler organism. Now, it's not these simple organisms, that's just joke slide, are organisms which can be manipulated in the lab. These have a smaller number of neurons. For example, the fruit fly has about a few tens of thousands of neurons, 20-ish thousand. Other organisms, such as the worm, which Sandhya Kaushika here studies, has far less. And it's possible for us to understand how these neurons are made, how they're born, how they grow, how they connect, and what is the mechanism which leads to their function. Now, the simple rules of development are shared between all organisms, starting from a fertilized egg, which divides to give 2 cells, then 4 cells, and 8 cells, and so on. And you have, for example, during development, a sheet of cells, sort of like us in this auditorium. And these sheets of cells, maybe at some stage of their development, be identical to each other. And then differences arise from them, which make them into different parts of the organisms. So an identical sheet can change the properties of its units in broadly 3 different ways. Cells can be in contact with each other. You can influence your neighbor, and your neighbor can become like you. You can influence a small neighborhood, or you can influence a larger neighborhood. This is called the American model, where your success depends on where you live. Cells can also change their properties depending on who their parents are. And if you're born to a particular set of parents, you are like them, and that's the English model, where heritage matters more. And quite often, cells can be identical to each other, and one cell randomly becomes different from the others and suppresses everyone from success, and that's the Indian model. So these 3 models of development are what, and these have been understood at molecular terms really well, what are the kinds of molecules involved. But don't take me seriously about these models and quote me. I hope this is not being telecast live and something like that. Yeah. It is? Okay. So, these kinds of molecules are used again and again in different contexts. And really, about 5 or 6 pathways which use these molecules are all what it takes to make cells different from each other. So groups of cells are similar. They signal at a short distance to make that block different from this. Then those signals within each other to create other kinds of differences, and so on and so forth. But the same toolkit is used again and again, typically within the organisms, to make immune cells, brain cells, skin cells, and so on and so forth. And the same toolkit is also used between organisms. Multiple organisms use that. So this shared toolkit at a high end is very important and common. And because of the shared toolkit, because location matters, and where a cell comes from matters, and where they end up or migrate matters, you make all sorts of differences in different parts of the body, including different parts of the brain. So that's a very long story on how this can be done. It's very exciting. It's an area where there's enormous amount of data coming in about understanding these systems better. But basically, it's a mechanism which uses a shared toolkit in diverse contexts in very different ways. And it's a big challenge to decipher that, but those tools for deciphering that are becoming more and more accessible and available. So you saw Leonardo da Vinci earlier, and here's another version of that, showing that, again, that you can study any organism and learn a lot about any other. There's, for example, a gene called Wnt, based on a fly gene called wingless, which is involved in many cancers. In the fly, its removal affects the wing. It was discovered in the undergraduate classroom in an agricultural research university, IARI in Pune, by Sharma and Chopra, and it really is an extraordinarily important gene for understanding how development takes place. So again, you don't know where value will come from in these situations. So that's how things are made. We learned about where they come from, where they're made, how does it work. That is physiology. And in terms of the nervous system, Hodgkin and Huxley took this giant squid, opened it up, took a giant axon from that, took the insides of the axon out and put in saline or various compositions there, and by altering currents, by clamping the neuron, as it were, could decipher what the properties of the neuron were, which gave rise to what we know is the action potential, the electrical signal, which propagates from one end of a neuron to the other. And this is absolutely extraordinary, because by just recording from a single cell, they could decipher and put together a set of mathematical equations which are consistent with predictions of specific molecules which are involved in setting up these currents and potentials which propagation. Today, it is possible to record from nearly 1,000 nerve cells at the same time, collect large amounts of data. Many of these channel molecules and also the molecules which are involved in connection, the synaptic molecules, all of them have been identified by recombinant DNA methods. So our ability to collect information about function of many, many neurons at the same time is absolutely extraordinary. So we have a path through evolution to understand where various components have come from. We have, again, through evolution and developmental biology, we know how these complex tissues, including the brain, are made somewhat. There's a lot to know in both cases. And we know from physiology and our ability to use fluorophores, molecules which tell us about the activity of cells at different times, and also from anatomy, which tells us about connectivity, how connection and activity relate to each other. Now we have to figure out how to put all of this together and how function emerges during development and in real life. So that's the last challenge. How is it that we have such extraordinary sensory motor control, some of us, to deal with life, and how is it put together during development, and what are the mechanisms of how this takes place through sensory motor and integration in real life. And there have been extraordinary studies, again, both in looking at different organisms from the outside, humans and other organisms, and looking at other organisms which are tractable from the inside to see how the acuity of the sensory motor system is put in place and functions. So this is something absolutely stunning. You have to have young Ronaldo going in for training, practicing to become as good as he is. He's not born with that ability. But a fruit fly, on the other hand, is born with this ability to play football right away at birth. Doesn't have to go to any training school or do any such thing. So how does this ability come about? Now, how development puts together this ability is related to a way by which the animal makes different components. It tests these components in different ways. For example, in the development of rat hearing system, rat ear, as distinct from, for example, in terms of visual system development. Visual system development requires at birth for the animal to experience different visual cues, and then the system matures. The hearing system, on the other hand, has fictive cues to mimic hearing before the animal emerges, is born, and therefore there's testing of that capability and ready to go when the animal is born. So there can be different kinds of maturity in different animals or different tissues. Mountain goats, for example, have this ability to run up and down the side of the mountain as soon as they're born. Some other animals will take a long time to be able to walk. So the basic tenet seems to be that you put together the right connections, you test the connections in fictive manners or in real life, and then you bring them out and you're ready to go. You don't actually have, in many cases, testing out the animal after it emerges or it's born, though of course there's a huge amount of maturation in different contexts which takes place. Now this again has been studied really well in multiple brain projects all over the world. And these projects, attempt to do things which were just unimaginable a short while ago. They chart out the connectivity in the entire animal, in the case of the fruit fly or the worm, or now people are trying to do that with zebrafish. It's a huge challenge. But these kinds of attempts to chart out entire connectivity of the brain runs into an enormous roadblock. The methods involved are electron microscopy of various kinds, and they can be rate limiting. They have to be both accurate as well as well done. Those technologies are improving enormously, but they are slow. But even when you have the EM sections, handling the data so that pixel by pixel you match between sections and do this segmentation analysis is a big challenge. There are major groups all over the world, big companies doing this through AI, so that's moving a lot faster, but not fast enough. And there are groups which outsource through large-scale crowdsourcing through students who actually do finish off and do a manual kind of completion of the segmentation analysis. And this has been done really well for the fruit fly, and the entire map of most of the fly's brain is now ready. So before this kind of segmentation, this is how it would look like on the left, and after that segmentation, you would get a much more detailed analysis. You have this wiring diagram of every neuron in the fly's brain, where it's connected, what the thousands of synapses it makes. But not only that, you can interfere with this wiring during development. You can record the function of each of these components in live flies, and you can activate or inactivate components. And you can look at behavior while you're doing this. So at least a documentation of what is happening everywhere, whether the input is one or multiple sensory inputs, is something which is extraordinarily feasible, and that's something which will now need to be examined from the perspective of hypotheses and mechanisms and how our understanding can improve. So this is an example of a completed annotation at very high resolution. So another factor which has happened is our ability to look at behavior in these contexts. Because you can manipulate every single nerve cell in these animals, you can do that and have these animals now moving around freely and see what they behave. And our ability to record multiple animals doing multiple things in different contexts, courtship, choice of odors which they like or don't, choice of food, mating, courtship, visual cues, all of that is just absolutely astounding what one can do at great resolution. And because of this mapping and because of this link of behavior to the inside of the organism, we can see from the outside and then also opening up from the inside what the organism is doing. So let's go back to the football fly. If you do this in slow motion and you label every point on its legs, you can see what it's doing. Because you know the position of every single muscle and every single motor neuron which innervates that muscle, from the outside you can say this is the muscle which is contracting or expanding. Then you can say, well, this is the motor neuron which must be coming on or switching off. You know where this motor neuron goes into the central nervous system. You know what other neurons connect to this, and you can start manipulating this and see what this happens. And you can invert this fly. It's now upside down on its back. You can put it on its feet, and it can move this ball, and you can show it a virtual reality of the world as it would be seeing it, and ask it to control that virtual reality. Keep some stripes within its vision, or go towards something which is attracting, go this other way, and see what neurons are doing what. It's really very, very impressive what that can be done. Now, that's impressive. So our challenge now going forward is this. Can we do this kind of work in larger organisms? What can we learn from doing this in fruit flies or worms or zebrafish even? What is it telling us new about the way the nervous system works? We have a checklist of all kinds of nerve cells, what kinds of neurotransmitters they make, how they connect, when they're firing, when they're quiet, what do they do at different times in development, when they think, when they sleep, when they remember. So that data is increasing at enormous speed. But what has changed about our fundamental understanding about how the brain works? We know a lot about the details of how cells work, somewhat about how groups of cells work, and what groups of cells are active or not at different times. But there's much to be learned. And one important question which many people ask, analogous to what the promises of the Human Genome Project was, that is such kind of large-scale brain ventures going to pay off or not, or is it a promise which is not likely to pay off? But in any event, it looks that the collateral benefits would be enormous, and the benefits in health, in declining, mental abilities, and in development, in how brain development takes place, there are already great benefits, and I think we're going to see a lot more. So let me end by showing you this picture. This is made by Sanjay Sane many years ago. And what we're trying to understand is how the steering, not the powering of flight, how the steering takes place, collaboration, which we've just started with Sanjay Sane and Michael Dickinson. And the wing in the fly beats by the muscles inside turning the thorax. And here, there's a banana in one of these four pillars, and the fly is going around. It exquisitely moves all over the place, and precisely on the wrong pillar, unfortunately, in this case. But it does, of course, many times choose the right pillar. So how does this happen mechanistically in terms of sensory feedback from the outside world? What are the sensory nerve cells on the wing and on the body of the fly? What's it telling? And the eye and smell, how they all come together to make the fly do this is very exciting, of course. But how it is made is something which is relatively, and lots of groups are studying that, but how it is made during development is something which also needs to be looked at, and that's what we're looking at, and also using comparative evolutionary approaches for that. So this is something which, as we learn more, we will probably know that we know less, and there are many tools to look at this. But right from how the brain is, where it evolved, how it is made, and how it functions, but also how the software of the brain is put together by us to result in massive cultural and social changes which we have brought upon this planet. We have moved from being on the planet of the apes to being on the planet of the apps. We have got an extended phenotype. We can do extraordinary things which other animals cannot do. How did that software ability come about is something which is very exciting, and I think this will be a lot of work which will go ahead on that. Thank you very much.
Thank you very much, Vijay. And I know you're all waiting for dinner, but I think some of you are even more eager to ask your questions. So let's have it open for 5 minutes.
Saying that there are more connections already made in the brain before at birth as compared with humans?
No, actually, newborn human, one of the immediate tests for functionality is to hold the baby up and it will do a walking kind of movement. That is then suppressed, people think, I'm not sure what the actual status of that work is, that the brain basically suppresses spinal cord movement in many ways until the brain itself develops better. So it's not that the development of the spinal cord and the motor system hasn't taken place. It has.
Just after, being born.
No, that's pretty impressive. A mountain goat or a horse can, run around soon after they're born. And how that system is functionally put together is extraordinarily impressive. We know, at least I'm aware of studies on how that happens in flies and so on and so forth, in maggots of flies and so on. But I don't know of studies which look on how that developmentally is put in place to have function over there. But its function is very impressive.
Yeah, but just after being born.
Yeah. Yeah, but a fly can fly when it's right after it's born. A dragonfly can mate in midair, which we need an aircraft to do. So they're very impressive insects. So this has been a topic of much debate. And that keeps getting rekindled when new tools become available. One, the collection of big data. The other tools are using deep learning, machine learning, AI, and so on and so forth. Brenner, in his article about a few years ago, I think it was on Turing's 100th birthday, is it possible? Yeah. He had a nice set of articles, it's worth looking at those. Basically, he says that, look, there are a set of people who bemoan the onslaught of big data and say that in our good old days, this wasn't the case. We had deep thinking and we used to come up with good hypotheses and solutions to problems, and now it's all collection of data. He says that's wrong, that you shouldn't bemoan the onslaught of data, you should welcome it. But you also need theory and an understanding of different domains in biology in depth to be able to link that into what is actually happening. And in some areas, this has grown enormously. In brain research, it is growing really well. There are overall, crudely put, there are two approaches. One is bottom-up, where you try to build circuits and get them to function, and brains and so on. That's informative. And the other is to break it down and see what happens, and try to see how machines can deal with these kinds of issues. So I think it's a new age, a new set of approaches. And as has happened in physics, for example, you're going to see now increasingly theory and experiment exchanging places in ways taking the science forward.
Sorry if I'm not phrasing the question correctly, but our attempt to understand the neurological processes and how the neurons interact and so on. So will it have a sort of a impact our understanding of the healing process? And then at some point in the future, is it likely that we will be able to simulate this healing process like the way medicines do at a chemical level, but can we teach the brain to sort of heal itself in whatever way?
Right. So there are two components. This is a very interesting point. One is, how does the brain, when damaged, heal itself? And there's an enormous amount of work which has been done in patients, in humans, who have various kinds of functional deficits or don't have it, to see what's happening. And it's pretty impressive to see how compensatory mechanisms can come. For example, there are compensatory mechanisms which come after strokes, after spinal cord injury, other kinds of injury. Recently, there was much fanfare about a study of a boy who, it turned out, was born without a cerebellum. The entire cerebellum was missing. And the boy is pretty much reasonably normal because, again, of what I mentioned, the extended phenotype, that family and friends and instruments compensate in many ways for what the cerebellum does. So these kinds of external prosthetics, just as we have hearing aids and spectacles and walking sticks and so on, these kinds of mechanisms of feeding back into brain dysfunctions will also be part of the healing. The principal problem in intervening for healing in the brain relates to this shared toolkit I mentioned. If every major item is used in every kind of cell, if you want to mess around with one part because there's dysfunction there, by putting an inhibitor or an activator of some component, and you're putting that drug in the entire system, you're messing it up everywhere else. So if it's a terminal illness, side effects being really bad doesn't matter because you can recover somewhat. But if not, it's a big challenge about effectiveness of drugs and drug delivery to particular targets. But there are very nice efforts now which can target not only drugs to specific groups of cells, but specific parts of cells, and then optogenetically or optically activate them in a manner which allows them to function locally. So again, there are new ways of healing which are happening. They're currently expensive, but I would keep in mind that there was a time about a few decades ago where a cochlear transplant, which would transform the lives of many people, was several $100,000. And now it's available for close to $1,000 in that range. So, and it's widely available, insurance companies will pay for it, and so on and so forth. And similarly, if you look at the history of spectacles, they are now very widely available for various kinds of purposes. So this is going to, as demands of an aging population increase, you're going to see a lot of interventions of this kind.
Okay, let's thank Vijay once again. And before we leave the stage, there's a memento for you.
Thank you. Thank you.